Hello students, welcome to SK Biology. Today we will study prokaryotic translation, one of the most important topic from molecular biology. Translation is the process by which the genetic information present in mRNA is converted into polypeptide chain, that is protein. In prokaryotes, translation occurs in cytoplasm and it is coupled with transcription due to absence of nuclear membrane. So, in prokaryotes, transcription and translation occurs simultaneously. I repeat, in prokaryotes, transcription and translation occurs simultaneously. To carry out translation, number of components are required. Let us understand all the machinery and their function. The first thing required is mRNA. In prokaryotes, mRNA is polycistronic, that is, it contains more than one gene. It is translated in 5' to 3' direction and its main function is it carries codon information. Second component is ribosome. In prokaryotes, 70 S ribosomes are used as protein synthesizing machinery. It is made up of large subunit, that is 50 S subunit, involved in peptide bond formation, and small subunit, which is 30 S subunit, responsible for mRNA binding and decoding. Third important component is tRNA. First tRNA in prokaryote is always formyl methionine, that is F-MET. Function of tRNA is it carries amino acid for polypeptide synthesis. Fourth component required is pool of 20 amino acid for synthesis of polypeptide chain. These amino acids added one by one as per codon information. Translation process also required some enzymes and protein factors to perform initiation, elongation and termination steps. In whole process of translation, some steps required energy in the form of ATP or GTP. Activation of amino acid requires ATP and elongation step requires GTP. Once all this machinery is available, translation process begins. Now, let us understand process of translation step by step. For protein synthesis, activated amino acids are required, which is carried out by amino acylation of tRNA. This reaction is carried out by amino acyl tRNA synthase enzyme. It uses ATP and adds amino acid to tRNA, forming charged tRNA. Once charged tRNAs are present, translation process is carried out in 3 stages: initiation, elongation and termination. We will understand each step one by one. First step is initiation. Initiation starts with interaction between 30S subunit with mRNA. In 30S subunit of ribosome, 16S ribosomal RNA is present, which aligns with Shine-Dalgano sequence of mRNA. Protein synthesis begins at start codon AUG on mRNA. Shine-Dalgano sequence is only present in prokaryotes. I repeat, Shine-Dalgano sequence is only present in prokaryotes. Ribosome contains 3 sites, A, P and E. A is for amino acid entry, P is for peptidyl transferase and E is exit site. Initiation process requires 3 initiation factors, IF1, 2 and 3. IF1 prevents binding of initiator tRNA to A site. IF2 has GTPase activity and directs correct positioning of tRNA. IF3 has function that it prevents premature association of small and large subunit of ribosome. After binding of initiation factors, initiator tRNA carries F-MET and binds directly to P site, not A site. A site is empty. This step requires GTP hydrolysis to GDP and it releases initiation factor 3, which leads to joining of 50S subunit to 30S subunit, forming 70S initiation complex. After formation of initiation complex, translation proceeds for next step, that is elongation. Elongation is largest and most repeated step. Elongation step also required 3 elongation factors, EFTU, EFTS and EFG. EFTU directs next tRNA to its correct position, that is entry of tRNA to A site. EFTS regenerate EFTU by GTP hydrolysis. And third elongation factor EFG mediates translocation. Elongation step starts with formation of binary complex in which TF-TU elongation factor binds to GTP. This binary complex convert to ternary complex by adding charged tRNA. Selection of charged tRNA is on the basis of next codon present in mRNA. Ternary complex is then added to the A site which brings second amino acid. After correct positioning of both amino acids, formation of peptide bond is done by peptidyl transferase activity. This activity is present in 23S ribosomal RNA present in large subunit of prokaryote ribosome. This step requires energy which is provided by GTP hydrolysis and GDP TFTU is released. TFTU is regenerated by TFTS for next use. After peptide bond formation, ribosome moves to next codon. This step is called as translocation, which is carried out by elongation factor EFG. Due to movement of ribosome, now A site is empty due to translocation. As per the presence of next codon in mRNA, next tRNA amino acid will come to P site and formation of peptide bond will be carried out in second and third amino acid. Every time uncharged tRNA is moved towards E site, that is exit site. So elongation step is subdivided into 3 cyclic steps. First is decoding, where selection of specific codon and anticodon is done, that is tRNA amino acid. Second step is transpeptidation, that is formation of peptide bond. And third step is translocation, that is movement of ribosome to next codon. These 3 sub-steps are repeated until and unless ribosome reaches to stop codon. Here it is UAG. Polypeptide is synthesized from N-terminal to C-terminal, and when ribosome reaches to stop codon, there will be no addition of any tRNA. Instead, it adds release factor. In prokaryotes, 3 release factors are known: RF1, 2 and 3. Any of the stop codon can be used by prokaryote, which is UAG, UAA or UGA. Release factor releases synthesized protein from machinery, and machinery is dissociated into 30S and 50S subunits and recycled for next protein synthesis. In prokaryotes, no any post-translational modification is needed. Prokaryotic translation can be inhibited with the help of different antibiotics, which I have shown here in this table.